Good morning, everyone. I am delighted to kickstart our 6th annual Single Cell Analysis Congress with our keynote speaker, Guilhem Tourniaire. Guilhem graduated with a Master in Applied Chemistry from Kingston University, later undertaking a doctorate in high-throughput chemical biology at the University of Edinburgh. After graduating, Guilhem worked for several startup companies, including Smart Holograms, Altrica, and Cellenion. This latest assignment led to the creation of Cellenion in 2016 and his appointment as Scientific and Managing Director of Cellenion, a Cellenion subsidiary based in Lyon, specialized in the fields of single-cell isolation and bioprinting. Guilhem will be presenting on cellenONE, an open platform for single-cell isolation and miniaturizing library preparation. Thank you very much for joining us today, Guilhem. Over to you.
So good morning, everyone. Thank you very much for the introduction. So I'm going to talk to you about cellenONE, which is a platform that we developed for single-cell isolation and nanoliter library preparation. Our company, Cellenion, was founded just about 4 years ago. We are based in France, in Lyon, and we are a spin-off of Cellenion, which is a 20-year-old company now, specialized in diagnostic manufacturing, which has infrastructure across the world and offices in Tempe, in Arizona, in the US. Our technology, cellenONE, is a bit peculiar as it can be used for two main applications, single-cell omics, but also for cell line development. Today I'm going to talk mainly about single-cell omics, so we are active in transcriptomic, genomic, epigenetics, and also single-cell proteomic, which is a growing field, as you will learn today. But we see a lot of interest for our platform for people who are developing new sequencing methods. It's a completely open platform, so you can really implement whatever library prep you want on this platform. In the field of cell line development, we are working with different companies developing immunotherapies, working on stem cell development, or doing gene editing with CRISPR-Cas9 applications, for example. The technology is based on side-drop Pico, which is an acoustic-based droplet generation. It consists of a glass capillary with a piezoceramic. What we have here is we applied a current, send an electrical pulse to the piezoceramic. This generates an acoustic wave inside the dispenser, which generates a small droplet. Droplets that are generated are highly reproducible and range in volume from about 50 to 800 picoliters, so tiny, tiny droplets. And then thanks to the reproducibility of this technology, we've been able to develop our single cell method, which is going to be explained in this animation that's going to start now. So inside our device, we have two imaging modes, one which is equivalent to a bright field microscope and the other one to a fluorescent microscope, and we combine those two modes. Inside the device, we can hold two MTP plates. This can be a variety of substrates, our new cellenCHIP, which I will tell you more about later on, nano-well structure like the cellenCHIP or 384, 1536 well plate. We really have no limitation in terms of substrates. We cool down those substrates in order to prevent RNA degradation, and we always start from a cell suspension. So you start from your suspended cells, you aspirate them into the dispenser, then we move you in front of a camera, and there we're going to image the cells that are in the sample. Basically what we do is we generate small droplets, and every time we generate a droplet, we take an image of the tip of the dispenser. And here you can see the ejection zone, which is the equivalent of more or less the volume of one droplet. So now, whenever we have a single cell in the ejection zone, we know that in the next droplets that we will generate, this single cell will be ejected. So we move to the target, eject one droplet containing the single cell. Go back to the camera, look again for a single cell event where we have a single cell in the ejection zone, go to target 2, dispense the single cell. And we do this until all the plate is filled. Admittedly, not all the droplets are going to have single cell. If they are empty, we discard them. If they have more than one cell, we discard them as well. And when we discard, we actually discard in a small centrifuge tube. So you have the possibility to recover all your sample and not lose any of the cells. All the cells that are isolated are imaged. We record the image. We take measurement of the cells. We measure the diameter, the elongation, and the circularity, and we use that to discriminate single cell from cell aggregate or cell debris to ensure that only single cell get dispensed. In terms of speed of the process, we can dispense about 40 cells per minute, so 100 cells every 2.5 minutes. We've now implemented the fluorescent imaging on the device, so we have 4 excitation channels, and we can now do most of the things that the fax can do. So we have DAPI, GFP, Sci3, and Sci5 channel. So you can do fairly complex sorting tasks. Here are some examples of what you can do with this. First example is just removing dead cells using a live dead stain. So in this example, the live cells are fluorescent green only, whereas the dead cells are green and orange. So we image first with the green channel, check the presence of cells. If those cells are only green, we move to the target and dispense them. If they are green and orange, we discard them as they are dead cells. That's what you're seeing now. This one is only green. And obviously, for every cell detected, we verify using the bright field channel that no debris or other non-fluorescent cells are present in the ejection zone at the time of dispensing. Now I'm going to move to another example. If you're looking for a cell subpopulation, in this example, the cells of interest are labeled with a red fluorophore. Every time we see one of those red cells, we move to the target and dispense it. And now we move to close to real speed. So we are half of the real speed, which is about 10 images per second. And you can see that you can cycle through a lot of samples to look for those rare cells and dispense them. And we have a final example that's going to come up now. In this last example, it's a feature that we've just developed recently, which allow what we call advanced sorting. It allows you to dispense different cells in different targets. So in this example, we have cells labeled in blue, some labeled in red. The blue cells go to the cellenCHIP number 1, whereas the red cells go to cellenCHIP number 2. In the event that we have 2 cells in the ejection zone, obviously we discard them. Now you're going to see, so in this example, we are dispensing different cell type on 2 different targets, so that you could imagine that you could do different preparation depending on the cells that you're isolating, and you're not limited to 2 groups, you can have as many different groups that you want. So this is for the technology. So in summary, it's very high accuracy. We can reach 100% of single cell isolated. It maintains very high viability, no stress on the cell. It allows you to inspect the sample. So if you have a lot of debris, you will see that in your sample, which is sometimes a problem with single cell experiments. We record everything, it's completely walk away. And as I said, it's an open platform. And we can process very small sample. Again, the main field of application. So now we have this little example here. I will try and use the pointer. So what you're going to see on there, it's a fairly old video that is 3 years old now. So it's an old version of the software. But what you see on the left is inside the device. On the right, what you see is the software interface. The tip is of the dispenser is here. The ejection zone is this zone here. So whenever we're going to have a single cell in this area, you will see the dispenser on the left moving to the target, which is a microscope slide here, dispensing the droplet containing the single cell and going back to the camera. I'm now going to start the video, and you will get a better idea. Here we are processing prostate cancer cells, which are very heterogeneous in size, as you can see. Some very small and some very large cells. Here a doublet, we just discard that, we don't isolate it. Single cell, and we do this as many times as you have position to fill or wells to fill. When you have 2 cells close by together, we discard the first one, only keep the second one. Here is the debris, we just discard that, and we do this, so on and so forth. Obviously, we can isolate mammalian cells, all sorts of mammalian cells, some small lymphocyte to the very large cardiomyocyte. We've done cells up to about 80 micron in length. But we can also do this for a range of different microorganisms. We've been isolating spores, microalgae, diatom, pollen. Obviously, we can do cell nuclei, and we are starting to work also in the field of bacteria. So these tend to be at the lower limit of what we can detect due to the low contrast that they have, but we are able to do isolation of those by labeling them. So we isolate them using our fluorescent module. So as I said before, you've now seen the single cell isolation application. And now I'm just going to describe a few more things that you can do with our device in terms of nanoliter dispensing for library preparation. So currently on the market, there are two main approaches that are dominating the market. Either people do single cell isolation with the FACS followed by preparation of libraries in microtiter plates. The advantage of this is that it's very flexible. You have many different protocols that you can follow. However, it's fairly low throughput. You're typically limited to a maximum of 384 cells, and you're working with large volume. And because of that, tend to cost a lot of money in terms of reagents, typically more than $20 per single cell. On the other hand, you have all the droplet microfluidic technologies, the 10X, the dolomite, the one cell bio. These have the advantage to work in nanoliter volume. They are high throughput. However, those technology cannot exclude doublets, and you get trouble when your sample prep is not very clean, when you have too many debris, and so on and so forth. And we believe that by using our cellenONE technology, we can get the best of both worlds. Basically, we have very few substrate limitation. We can dispense in virtually any type of chip, as long as the wells are going to be larger than 50 micron in diameter. We can work in picoliter to nanoliter volume range. We can undertake multiple interventions, so we can do many type of library prep. We can correlate the images of the cells that we are isolating to the sequencing data. It's automation friendly. But the main challenges that we were facing when wanting to do this is that there is no commercially available substrate to do that. So we had to develop our own substrate. Our US team actually was implemental in the development of those chips. So these are called cellenCHIP. They consist in 4 arrays of 96 wells, so 384 position for single cell onto the format of a microscope slide. You can do reaction in volume ranging from 20 to 500 nanoliter, and you can have up to 8 cellenCHIP onto the deck of our device, so up to 3,000 single cell isolated per experiment. So once we developed this chip, then we wanted to test what it was like to prepare libraries in those. So the first thing that we did is we went out, we purchased one of those commercial kits. So we purchased the kit from QIAGEN, which is a 96 well plate based kit. And basically what we did is we implemented this kit onto our cellenCHIP. And by doing so, just by adapting exactly the same protocol, we were able to reduce the volume of the reaction from 5 microliter per well to 100 nanoliter per well. And the results we obtained were very comparable to the 96 well plate results. However, we were quite disappointed with the quality in terms of number of genes detected per single cell, which was just below 2,000 genes. So admittedly, we managed to do a drastic cost reduction, going from $35 per cell just to $2 per cell just by using those chips. But we wanted to obtain better results. So we got our molecular biologist team to work, and they tried to implement an scRB-seq protocol onto the cellenCHIP. The advantage of this protocol is that it allows us to remove one step. So it consists just in barcoding of the chip. Then you place your RT buffer together with your lysis buffer. Then we do the single cell dispensing. And you do directly your RT, and then you can pull into those funnels that we have developed that are suited for this cellenCHIP. As you can see, the results we obtained were much better. We tested 3 different RT enzymes, and you can see that without even optimizing the protocol, we were able to capture, on average, more than 5,000 genes per cell. And we are currently working in optimizing this protocol to capture even more genes. So if you are interested in implementing the work you're currently doing, maybe in microplate, onto this chip, don't hesitate to talk to us. On the right, you have this QR code that will take you to a questionnaire. You can fill in your detail, and we'll be pleased to discuss with you what you would like to implement on this platform. As I said, it's a completely open platform, and we are really pleased to be able to work with you to implement new protocols. So now I'm just going to give you a few examples of work that has been done using our device, mainly by our customer on the field. So I will give you an example of genomic, of transcriptomic, and an example of proteomic work. So the first example is a paper that was published last November in the journal Cell. This is a method that has been developed at the British Cancer Research Centre and UBC in Vancouver. It is called Direct Library Preparation Plus. It is a very nice method for whole genome sequencing. It allows a number of parameters to be determined, copy number variation. You can do clustering, you can do single nucleotide variation, reconstruct lineages. This has found a lot of application in cancer research. This work was developed by the Professor Sam Aparicio and Professor Saurabh Shah. And it provides very nice high quality data with very low preamplification. It's very versatile as it allows analysis of cells, of any type of cells. It's fairly low cost with a few cents of dollar per cell. And it is now, this method is now being implemented as a routine method at the British Columbia Cancer Research Centre. And they've now analyzed, well, that was like 6 months ago, they had analyzed over 400,000 single cells with it. And this method is now being implemented at research centers all over the world. It is being used at the Francis Crick Institute in London, at the Memorial Sloan Kettering Center in New York, at the Japanese Foundation for Cancer Research and the Children's Cancer Research Center in Sydney. So very nice method. I really encourage you to have a read through this article. Outstanding work there. My second example will be in the field of transcriptomic. This is work done actually at Cellenion in Lyon in collaboration with the Cancer Research Institute in Lyon with a group of Professor Léa Payen. We are trying to isolate single circulating tumor cells to do transcriptomic analysis. I don't know how much you know about circulating tumor cells, but these are believed to be the seeds of metastasis. And as you probably know, metastasis is typically the killer in cancer. So there is a real need for new methods to isolate single circulating tumor cells to be able to better understand the response of patients to drugs or to a treatment and to find new treatments for those patients. But the main challenge working with circulating tumor cells is that they are really, really rare. So to give you an idea, in a typical patient, you have about 1 circulating tumor cells per 10 billion of cells in the patient's blood. So as you can imagine, finding those CTC, it's like finding a needle in a haystack. And another challenge linked to those circulating tumor cells is that they are very heterogeneous, both in size and in the marker that they are expressing. So there are different approaches to enrich those. Some methods are based on physical properties. They typically select the CTC by size, as they tend to be larger than the blood cells. However, some CTCs are known to be as small as lymphocytes, so you will miss those if you do such an approach. Another approach is using immunocapture. However, as I said, they tend to express different markers and they tend to undergo the epithelial to mesenchymal transition. So it's very hard to find markers that are going to capture all those. So typically, those enrichment technology will achieve a log 5 to log 7 enrichment. So you don't end up with a pure CTC sample at the end. You typically end up with a sample that contains about 1 CTC for 1,000 to 10,000 cells. And with such sample, there is not that many methods that are really well suited to do single cell analysis. So here is a short video of the technology we are using for enrichment. So it's a company called Biolytics that has developed the ClearCell FX1. It's a label-free technology that allows automated and fast enrichment. As you can see here, the larger cells are collected into a side channel. One of the main advantages is that it is label-free and the cells remain intact, which is essential for transcriptomic analysis. And here is a description of the protocol that we have developed. So we start from patient sample or spike sample with about 1 CTC in 10 billion cells. We enrich those samples, it takes under 1 hour. We obtain the enriched sample with about 1 CTC in 5,000 cells. And there you cannot do a 10x experiment, otherwise it will cost you a fortune because you will have to sequence, let's say, 10,000 cells to find 1 CTC. So you really need to find a way to really capture the single cell. The FACS won't be able neither to isolate those, as there will be too few events for you to set the gating. So our approach at this stage is to do an immunostaining. We label all the white blood cells, and then we do a negative sort. So basically we isolate everything that is non-fluorescent, and we ensure that there is no debris or no other cells using the bright field imaging of our device, the IPSI technology. And we dispense single cell into wells. We use a next kit in this collaboration, as New England Biolabs kindly offered their collaboration in this project, and we then do the sequencing. Doing this, so we started the work by doing spike sample using this HMEC cell line, which is a human mammary epithelial cells. These cells are a very good mimic for CTC because they do undertake this transition. And actually, we were able to demonstrate this in the first experiment that we run, where we saw that the single cells that were isolated were showing this very heterogeneous phenotype. So we've developed a nice protocol combining the ClearCell-FX1 with the cellenONE F1.4. The negative immunostaining strategy that we implemented is showing very nice results, as we were able to demonstrate up to 87% recovery of the spike cells in the sample. The library prep is working very nicely as well. We generate full-length transcriptome data. We've now implemented the freezing step after enrichment. This allows for a much easier logistics, so that once you do the enrichment, you can freeze your sample and then do the single cell isolation in a second step on another day. And we are now processing patient sample and preparing the manuscripts that will be published in the coming months. I would like to thank on this work Professor Léa Payen, Jessica Garcia, who was a PhD student in our group, and now she's a postdoc there. François Monjaret, who's a colleague of ours working at Cellenion, Biolidics and New England Biolab for their collaboration in this very nice project. Finally, I will just present in a few words some work that has been done in single-cell proteomics by Konrad Lohr at the BAM Center in Berlin. So their work consists in using laser ablation ICP-TOF-MS to undertake single-cell proteomic analysis. So they use different strategy. In the first step, they were using markers labeled with metal ions in order to detect and quantify proteins and the quantity of DNA present at the level of single cell. And their method is also suitable to detect the presence of metals with very high detection limit, very low detection limit, sorry, at the single cell level. But so I will not say much more on the single cell proteomics, because I know that in about 1 hour time, Nikolai Slavov, who's an outstanding presenter, will present his work that is being done at the Northeastern University in Boston. So in summary, our cellenONE technology offers some unique features for the omics field. It provides very high single cell accuracy and certainty. We only isolate single cell, no doublets, no debris. It's versatile in terms of cells that can be used, and you can use anything up to about 80 micron in diameter. You can use the technology for nanoliter library preparation. So if you have any new type of library prep you would like to miniaturize, please don't hesitate to come and talk to us. I didn't demonstrate the application there, but we are really good for rare cell isolation. Well, actually CTC was an example. We can work with just a few microliter of cell suspension, and we can achieve outstanding recovery rates up to 95% in a single pass. We visualize the sample so you can get a real idea of the quality of your sample prep, which is critical when you're doing single cell experiment. We record all the images of the isolated cell. And as I said, it's a completely open platform. So if you want to try something new, just come and talk to us. Finally, all the stuff that I presented wouldn't have been able without our current customer and partners. So we are working with a number of prestigious cancer research centers all over the world, a few in the US, in Europe, also in Asia and Oceania, as you can see. And we have our system installed at many sequencing platforms at universities. Finally, I would like to thank you. If you have any question, you can contact us directly on our website or at contact@Cellenion. And on the Oxford Global app, we have a number of colleagues. Their name is there, or you just type Cellenion. If you have any question, we'll be pleased to get in touch with you. Thank you.
Thank you, Guilhem, for a great presentation. So as Guilhem kindly mentioned, he will be available via the app if you would like to reach out to him there, or as he said, on the Cellenion website. So thank you, Guilhem, again for a fantastic presentation.